Actually, my task today was to talk about Dr. Warner Greene's contributions as an HIV virologist to COVID-19. And I started looking into it, and I was amazed that it's only been 3 years of COVID and how many contributions, given, I think, Warner's longstanding knowledge of virology, immunology, inflammation, how much he's contributed to COVID-19. So I'm going to go over some of those. Yeah, that needs to be done. Thank you. Thank you. That's good. How many contributions he's made to the effort of COVID. There's no doubt that these are the two great viral epidemics of our day. And if we think about where we are with HIV, we're not where we want to be. So 38.4 million people living with HIV worldwide as of the end of 2021, 40.3 million deaths from HIV/AIDS since the beginning, and really first described on June 5th, 1981. And you've already heard the contributions of Dr. Greene and Dr. Gallo and Dr. Deeks and everyone in this room to the description of HIV. And then COVID-19 it's sort of symbolic because March 9th is essentially when the global health emergency was declared yesterday, 3 years ago. And then the pandemic, declared as a pandemic, is tomorrow, March 11th, 2020. So we're now 3 years in, 6.87 million deaths since the beginning of the pandemic when it was declared. And there were many people who pivoted from HIV and worked on COVID, and I was really impressed by what Warner has done in that area. And I think it's really three main contributions that I would kind of put together is how Dr. Greene's expertise in HIV translated to pivotal COVID-19 work. I think the first one is immunology, and Dr. Gallo mentioned this, but T-cell immunology is terribly important in HIV, but incredibly important in COVID-19, and really pivotal papers, which we'll go over, that Melanie Ott, Nadia Roan, and Warner Greene, and the entire group did on defining T-cell immunology responses to SARS-CoV-2, how they're both helpers and hurt and cells that hurt, because there's an immunopathology aspect of SARS-CoV-2 that really got defined by this group. A lot on virology and transmission, really in the area of mother to child and placental transmission, which we'll talk about, and then really recent contributions in inflammation and long COVID, post-acute sequelae of COVID. So let's talk about those. This was March 27th, 2020. So, right, this is only 2 weeks afterwards, and the San Francisco Public Press put out an article that this lab, Warner Greene's group, was going to essentially pivot to work on battling COVID-19. And the article was specifically, frankly, about hydroxychloroquine, but that wasn't the point. Like, the point was that, we didn't know. But the point was this idea of very rapidly figuring out through the infrastructure of the AIDS research enterprise and through Gladstone and through Warner Greene's lab, thinking about treatments, thinking about basic mechanisms of COVID-19. And that work continued, and it was started so early. So what about, and then before we get to the specifics of this work, there was a really good communication from Warner to the press and to the media. And I think these were two incredibly prescient interviews, because remember that the first one was in MedPage Today, and this was February 2021. So this is when the vaccine was starting to be rolled out in this country. I think healthcare workers had received it before Delta, and we didn't know what was going to happen. And very presciently, Dr. Greene said, yes, the virus still has tricks up its sleeve. And indeed, Delta variant appeared, first described in India, but appeared and became the dominant variant in the United States by July of 2021. And then this article from USA Today where Warner commented about Omicron. And this was because of his extensive knowledge that Paul just talked about of understanding research that was going on in Africa, really believing South African scientists and clinicians who were saying there's something different about this. It's milder. Maybe it's population immunity. Maybe it's less virulent. But whatever it is, this is different. And to be able to harness that experience of working with South African scientists and believing them, and essentially saying, his quote, "This looks to be a highly infectious virus, but it may not be as virulent or as pathogenic as the Delta variant." And that's exactly what played out. So let's talk about, so these are prescient interviews before and during each variant, but let's talk about the specific papers, the specific work that Warner did with his group on talking about how to define COVID-19 pathology. So the first stop is T-cell immunology. I learned a lot about T-cells during this pandemic, but I learned it from this group. It really is about how important, of course, the T-cell response is, and the Th1 specifically T-cell responses for intracellular pathogens. And so this was known. It's not like we didn't know this about viruses, but how do we play this out? What was happening with SARS-CoV-2. And there are 4 major papers that Warner and his group published along this idea of what the T cells were doing, both in immunopathology, and then what were they doing either from infection or from vaccination to combat SARS-CoV-2. And these were in, unshabby journals like Nature and Cell. And so what are the pivotal findings of these papers, really? This work is that, of course, SARS-CoV-2 T cells have a pivotal role to play in the control of COVID. But prior to the development of immunity, whether it's from infection or whether it's from vaccination, this increased proportion, again, working with our HIV expertise, these increased proportions of inflammatory CXCR4 positive T cells were found in the lungs of patients who had severe COVID-19. That's the contribution to the immunopathology of COVID-19, that lung-homing T cells are activated through bystander effects, and they contribute to the actual disease itself prior to the development of adaptive T cell immunity. And then where does adaptive T cell immunity come from? And of course, it comes from the vaccine, or it comes from infection. And both infection and vaccination generate strong T cell immunity, and that T cell immunity has diversity and it has breadth. And then one of the most important papers, I think, is really shown here on the left, but actually the top left, but the importance, as universally scientifically agreed, that vaccines were our way, to generate strong T cell immunity. We don't desire people to get infected to get their T cells. So this is an incredibly important paper in Nature. Okay, what about the second question, which is virology and transmission? So again, harnessing significant expertise from Warner Greene to turn it towards, okay, what do we know about transmission, specifically focusing actually on the important transmission questions of the placental transfer of SARS-CoV-2. And this is important also to allay myths, because I'm going to tell you about a paper that the group contributed to that allayed the myth that there's going to be products from the mRNA vaccines crossing the placenta, which is a commonly kind of used concern in the misinformation aspect of COVID-19 vaccines. So really 3 major papers here. Again, JCI, Nature, Communications were really the major, I think Dr. Gallo said this, but these kind of very high-level journals is why there was such an impact from the work that Warner did during COVID. And so we'll talk about the work on SARS-CoV-2 can infect human embryos, and then we'll talk about the transfer of antibodies across the placenta during pregnancy. And so this was that first paper mentioned here on the upper left. This is the paper about the actual infection of embryos. This was hard work. These were actually discarded human embryo work. So it was the question was, could there be direct infection of SARS-CoV-2 of the human embryo? And really impressive work that these pivotal receptors, cell entry receptors for SARS-CoV-2, ACE2 and TMPRSS2, they were actually expressed in cells on the embryo, on these pre-implantation embryos. And so by definition, then there was robust infection by SARS-CoV-2 of the trophoectoderm cells in the embryo. And the reason that that's so terribly important is that it's incredibly important to have immunity and neutralizing antibodies around during pregnancy to combat infection, to prevent infection of the embryo during COVID-19 infection. So that's the next two papers that were published that really looked at paired maternal and cord plasma samples from pregnant individuals. Pregnant individuals, 30 were vaccinated, 30 had immunity from natural infection, and importantly, neutralizing antibodies generated by either of these two states were pivotal to preventing infection and essentially absolutely can cross the placenta and protect the fetus from SARS-CoV-2 infection. There were some nuances about when you want to vaccinate. If you vaccinate in the third trimester, that actually induces higher neutralizing antibody titers at delivery, but then they're actually a little bit lower later. It turns out the best time to vaccinate is late in the second trimester, but point is that this is like incredibly clinically meaningful work. And then really important as we combat kind of misinformation, combat myths about the mRNA vaccines that have been really plagued our ability, I think, in the United States more than any other country to truly combat COVID-19 with our lower rates of mRNA vaccination than so many other high-income countries. Really important to show that there was no evidence of the mRNA vaccine product going across maternal blood, placental tissue, cord blood. These, as you would expect, the mRNA from the vaccine quickly degenerates and causes zero damage. So I think that's incredibly important work as we're like working towards increasing trust in the vaccines. And then the final, and I think this is still happening, and Warner told me outside I'm not supposed to say, but I think there's going to be some really important, like, big paper coming out. But all I know is that what I can see in PubMed, and essentially two really important papers on inflammation and post-acute sequelae. So those two papers are here, and the bottom one is a preprint, and it kind of looks like it could get into Nature or something. But anyway, the top one is really published in the American Society for Microbiology, and then the bottom one is still in preprint, but I think it's going to go somewhere big. And really, what are these two papers telling us about kind of the role of SARS-CoV-2 in generating inflammation? That is so up Dr. Greene's sleeve, because that's what he's been working on so much in terms of the HIV response and inflammation. So just that incredible pivot to COVID-19. So what were the findings from the group from the neuro paper? Well, it's just really showing that astrocytes are directly infected by SARS-CoV-2. That's terribly important. Remember, that happens with HIV. Remember, they don't affect neurons, and there were all that kind of inflammation around the neurons from infection of other types of cells. And this leads to reactive inflammation, just essentially inflammation. And now we need to figure out how to connect that clinically with long COVID work. And then this is a very important preprint, but this preprint really shows this question about what is the mechanism by which clotting can be stimulated by SARS-CoV-2 infection. And this paper really shows us that the spike protein from the virus itself binds, not from the vaccine, binds to the blood coagulation factor of fibrinogen in a mouse model. And that induces two things. One, going back to the immunopathology aspect of this disease, it induces fibrinogen-dependent lung pathology. So that's why immunopathology is so important, and that's why we use steroids. And there's just so much about immunopathology with this disease. And then afterwards, if there is a higher level of the induction of fibrin deposition, can we think about using fibrin targeting interventions in treatments? So really, really important work. And I don't think I knew it because I had seen him in the news, but I just did not know, how, I think if you think about those three areas, how pivotal all three areas are to, number one, kind of increasing trust in the vaccines and the importance of the T-cell response. Number two is increasing trust in the vaccines and the importance of vaccination and immunity during pregnancy. And then the third is basic mechanisms of inflammation. So I'll just end with, we are all in this room because HIV isn't over. And I do have to come back with our remembering of what also happened during COVID-19, which were setbacks in the HIV response. And that's important because I think we do have to do two things at the same time. And we are now, again, at a point where the UNAIDS update that they give out every 2 years, usually kind of perky titles. This last title was In Danger. And that's where we are with the HIV setbacks. 40.3 million people have died. 38.4 million are infected. 650,000 deaths. Every minute that we talk, someone died in 2021 of AIDS. Really high rates that actually worsened in adolescent girls. Every 2 minutes, an adolescent girl or a young woman is infected with HIV. That worsened with the school closures and with everything else that happened worldwide. And still 75%, you could say that 75%, or you can say that 25% of adults don't have access to ART. And 48% of children don't have access. So we're not even by a long shot done with HIV. And I think we do have to pivot back and forth between the two if we decide to work on COVID-19, because this isn't over. And then it has been really my privilege to have inherited the CFAR from Paul and Warner, and we inherited it in amazing shape. And we were really trying to compete because they would do renewals and they'd get scores of 12 and 14 and 11. And so we were really trying to compete. We did okay, but we we had a lot to compete with. We got a 10, but we had a lot. We had a lot to build off of, to compete with, to work off of, and it was an incredible CFAR to inherit, and really mine and Peter's and Mallory's privilege to be in leadership of the CFAR from these two great men. So thank you so much, Warner, for everything you've done. Thank you so much.